Thank you very much, Camille. I'm very happy to be here and to share with you some phenomena, collective phenomena that we found in transport systems, which I find quite fascinating. So actually, my talk will have 3 parts to show you examples on 3 different systems. First, I will speak about intracellular transport, so transport that occurs inside cells. Then we'll go to pedestrians, with human pedestrians, and then to road traffic, to give you a flavor of the kind of collective phenomena we can meet in these systems. First, for those who are not familiar, why do we need transport within cells? A cell is not something homogeneous. You see here a cartoon. You have different compartments corresponding to different functions. Basically, a cell is like a town. You must have some sources of energy. You must have some synthesis of proteins, a number of things. You must deal with your waste. You have exchanges with the external world. And you may have to grow or move your cell. And this occurs in different places. So you need some logistics to have your city work well. This is all the more true in neurons. Neurons have a long extension here called the axon, which in the human beings can be up to 1 meter. And so you really need transport within this very, very elongated parts of the cell. This cannot be done by diffusion, given the dimensions, but also the fact that it's very, very crowded. Here you see a small part of the axon. It's full of all kinds of filaments, of vesicles, mitochondria, and everything. So you need really active transport in these systems. How is this done? You have some roads. Here is the network inside the cell. So in blue, it's DNA, it's a nucleus. Then you have some filaments radiating from the center to the outside of the cell, which are the microtubules. So microtubules are polymers like stiff cylinders, which are somehow the highways in the cell. And then you have another kind of network, rather concentrated near the external wall, made of actin, which are also polymers, but not so stiff. So I will mostly concentrate on transport on these microtubules. So now how do we transport something along these roads? There are some molecules called molecular motors, which really have kind of legs. And these molecules can perform steps. Of course, these steps are very small. It's, for some of them, 8 nanometers. But they can go very fast. If we would be these molecules, and we would just scale up, we would walk at a speed between 200, 300 kilometers per hour. So they go very fast. And these molecules are responsible for this active transport inside our cells. Something very important is that microtubules are polarized. So imagine you are on the microtubule, you have tiles on the ground, and just by looking at the tiles, you know that Paris is in this direction and there is Marseille. So everywhere you can choose in which direction you are walking. And you have some motors specialized to go to what we call the minus direction, and some others specialized to go to the plus direction. So we can go in both directions on these highways. Now it's a bit like science fiction. When you are walking on the road, sometimes you can detach, diffuse around, and then attach again before moving on. So this seems quite simple now, but there are some questions. First, you could imagine that when you want to carry a cargo, you would attach one motor of one kind, and then you would go in the direction where this motor walks. But as I said, the system is quite crowded, and you may have an interest to attach several motors to be able to apply stronger forces, and also to increase processivity. This means that if one motor detaches, the other one would not detach at the same time. So you would stay attached to your microtubule for a longer time. So all this seems favorable. But as I said, you have two types of motors. And so there are some questions. Do motors just attach randomly? And then you would expect that you have the two types of motors attached at the same time. Or you could have some external factor, whatever, that would prevent one type of motors either to attach or to walk, but they would somehow decide who is driving the cargo. It may depend on the system, but there are a number of evidences that I will show you some of them that we are more in this setup. So having motors of both types attached to the same cargo. An example, here we see the motion of an endosome like a vesicle inside a cell. And first you observe that they go in both directions. So the motion can be reversed. But more interesting, when the motion is reversed, so this is position and time. So it goes in one direction, then in the other direction. And you have a fraction of time where it's like hesitating. It's just stopping. And if you look at the endosome during this period, you see that it becomes elongated. So here is the length of the endosome. You see that during this period, it's getting elongated as if teams were pulling on it from both sides. So people came with the idea that it's a bit like this game, the Tug-of-war, where you are pulling on a rope with two opposite teams. And you know that this is not a very efficient transportation system. It doesn't move usually, not so much. But if the cell chose this, there must be a reason. So that will be my first mystery. Why is it so? We'll try to understand this. So we want to understand what is the dynamics of a complex composed of one cargo plus two teams of motors pulling in opposite directions. We want to know if we can compare something with experiments and if we can predict something and understand what's going on. So I will start with a mean field model, which was a historical model that was quite famous because it gave a first hint that we could understand why it is so. In this model, so you have the cargo, and motors are attached. And actually, the picture is a bit misleading, because in this model, you do not describe where the motors individually are attached. You just say, OK, I have two blue motors and three yellow motors attached. And in each team, you make an assumption that the blue motors which are attached, they share the load. So they pull with the same strength. That's an hypothesis. So just by knowing how many motors are attached, and with this hypothesis, you can make a model where you describe the motor dynamics. It's you have some detachment rate that depends on the load, on the force exerted on each motor. They can move with a certain velocity, depending again with the load. And they can also, once they are detached, they can attach again. If you do this, you find two types of behavior. If the motors stop walking before detaching, because, yeah, I should say, when the motor walks, if you have a force pulling backwards, above a certain force called the stall force, it will stop. It cannot move forward. So, and also, if you pull stronger, you detach more. So if you are in a regime, in a type of motor, where they would stop walking before they can be pulled up, everybody is just stopping and nothing moves. So you have just a velocity distribution, it's just a peak around 0 that corresponds to the tug-of-war game where mostly nothing moves. But if you are in a regime where motors can detach before they stop. Then one team manages to pull off the other, and then the cargo can be taken away with a very high velocity, which corresponds to the velocity of a free motor. So this would give you a bimodal velocity distribution, each peak corresponding to one direction. And you have the intermediate case where you have the two situations. So the mean field prediction was, yes, with a tug-of-war, just by random fluctuation, you can obtain transport, and very efficient transport, just by pulling off the other team. So we were there when we started. So this is a collaboration with Ludger Santen in the University of Saarland. And we are collaborating by having some joined PhD students. So what I will present today is the work of Sarah Klein, who finished her PhD last year. So she developed a model inspired by previous models, where you represent explicitly the position of each motor. So now you know, so the yellow motors go on the left, the green go on the right, and you know the position of each motor. They are linked with a spring to the cargo. So now when you know the position of the motor, you know the force it exerts on the cargo. And it's different for each motor. It's not, we are outside of the mean field assumption. They feel individual forces. And the dynamics is the following. You have a stochastic dynamics for the motors, where they would attach, step, and detach with some rates. And these rates depend on the force that they feel. And then you have the cargo dynamics, where you have a friction force due to the surrounding medium. And you have the sum of all the forces exerted by the little motors. So you have two coupled dynamics. Now each time the cargo moves, it changes this individual force because it's just a spring. So when the cargo moves, it changes the relative position between cargo and motors. You had a question?
If the motor can cross on the microtube?
Yes. Actually, we checked both. It will not change qualitatively my results. We chose to say that they can cross because here we have a single filament, but actually on the microtubule you have parallel tracks, and they could somehow be on parallel tracks. So we have no exclusion, but we have checked by putting some exclusion that it will not change the conclusions. So now once we have this model, we try to see if we still have the results predicted by the mean field model. So in the mean field model, in this picture, it would correspond to have all the motors attached at the same position because they feel the same force. They share equally the load. So it means they are exactly at the same position, and they all step together, which this is to help you realize what is behind this assumption. While in our case, they have all different positions. While in the mean field model, you have so this bimodal or trimodal velocity distribution. When you go to the explicit model, this is lost. And it's not just a matter of changing the parameters. You don't get it. So it seems that these high motility states defined like this are really an artifact from the mean field assumption. So one way to understand why it is so is that if you are in this configuration, when one motor is pulled off, the force immediately increases on the other ones. And so they are more likely to be also pulled off, and everybody goes away at the same time. While when you are in this model, so this is the space on time, and you see the cross is where the motors attach, and then they step. So the red go upward, the green go downwards. And the color here gives you the ratio of the detachment rate above on the stepping rate. But it's mostly the the force applied on the motor, if you wish. So when one motor, for example, here detaches, you see that the force does not immediately become very high on the other ones because they are not at the same position. And also the cargo will relax its position, so these ones have time to still continue walking and take over. So it's much more difficult to pull off one team. So you may wonder, then we are back with the problem. How does it work? So in order to compare with realistic experiments, we have to go a little more closer to reality. Until now, I considered symmetric teams to be able to compare with this mean field model. But some important information is that the motors are not, the two types of motors are not symmetric. So these families are mostly called kinesin and dynein. And for example, for the detachment rate as a function of the force, so this is for kinesin and dynein, they behave quite differently. In particular, above stall, look, here, the detachment rate increases with the force. So when you pull harder, you detach more the motor. But for dynein, it's the contrary. When you pull more, it gets more anchored and it doesn't move away. So this is called catchment behavior. And so it will make a very different behavior.
Sorry, I missed. What is the difference between the two? No, I understand the graph, but what is going on in the two cases, I missed.
I mean, the kinesin is a type of motor going in one direction, and the dynein is a type of motor going in the other direction. So somehow, is it your question?
No, I'm missing what these two graphs are, what physical situation they're representing. That you have more motors of one type than the other, or what?
No, in this, typically what you do, it's in vitro experiment. So you put a microtubule, then you put a motor on it with a bead, and you exert a force with a magnetic trap, for example, F, and you measure the detachment rate. And you do this for one kinesin motor and dynein motor.
Oh, so just one is a test?
Yes, in this it's the property of one individual.
Okay, and one is going opposite to the force you apply, and the other one, is that?
Oh no, in both cases you apply a force opposite to the motion.
And it's two types of motors.
It's two types of motors, yes. You have also different properties for the stepping rate. So the stepping rate, I said, it depends on the force you apply. It also depends on ATP concentration, and it's quite well fitted by Michaelis-Menten kinetics. Here you see, for example, for kinesin, the velocity as a function of ATP concentration is quite well fitted by this law, and for velocity as a function of the force, again. So we know quite well these things, a little more for kinesin than for dynein, I should say. But still, we know that they are quite, the parameters in this law are quite different for kinesin and for dynein. So you have motors with different properties. Okay, we'll see what we'll do with this. But so now we will observe how it behaves and whether we can compare with some experimental facts. So some experimental facts that people measure are anomalous diffusion. So just one short remark. What people measure is the mean square displacement. So for a ballistic motion, you would expect an exponent 2. For purely diffusive, an exponent 1. And if you are below 1 or between 1 and 2, you will say it's anomalous diffusion. So the first remark is that when you measure this for finite time, actually it's a bit ambiguous, because when you find an exponent between 1 and 2, so you speak of superdiffusion, actually you don't know if it's really superdiffusion or if you just have diffusion, that is uncorrelated motion, plus a bias. And as I said, the two teams are different, so you expect that the generic situation is that you have a bias. There is no reason why it would not be biased. But OK, that's what people do. In our numerical studies, we rather take the variance to make sure it is really superdiffusion. So we remove this bias. Okay. Now in the experiments, for example, you put beads that you manage to internalize in the cell, and they are coated in such a way that they will be transported by motors along the microtubule. And you measure this mean square displacement. So it corresponds to this black line. And what you observe is that you have a regime with superdiffusion, exponent 3/2, which occurs. So please remind a bit the transition. Here, it's about the scale is seconds, so a few seconds. And then above, you have actually either here it's subdiffusion, but you can also have just pure diffusion. It depends on the size of the sphere. And this you can understand if it's very crowded. Small spheres can diffuse, and the big ones, they are a bit hindered by the obstacles. So we somehow will not care about this, which is more related to what is around. But we are interested in this superdiffusion. Here it's just for comparison. If you have granules which are not driven, you find the subdiffusion due to the obstacles. So it's really due to the driven motion that you have this superdiffusion. Another experiment now in vitro, you somehow crush an egg, and then you use this. You put beads coated in motors, and they are moving along microtubules. And again, you find that in the presence of microtubules, you have this superdiffusion with an exponent which is about the same. If you remove the microtubule, but you leave the actin, so you leave the obstacles, then you find the subdiffusion. And if you have no filaments, it's just diffusion, as you expect. What is interesting is the comment you find in this paper, which is quite representative of what people were believing at this time. They say, OK, we find some superdiffusion, but it comes from a hindrance to ballistic motion. So it means people were thinking this cargo motor complex, it should move ballistically along the microtubule. Well, we find only superdiffusion. This must be because there are some obstacles or something. So they have the belief that the motion must be ballistic. Another example, now we follow peroxisons which are naturally present in the cell. Again, we find this superdiffusion with an exponent close to 3/2. So you see now it's milliseconds here. So the transition for a few seconds here, it's 10 seconds. And there you have another transition, which is more around milliseconds, where you are subdiffusive. So you find actually 3 regimes. In the other experiments, we didn't reach these scales. We couldn't see it. But so for very large time, it depends on what is around. You have diffusion or subdiffusion. Between seconds and milliseconds, you have superdiffusion. And now, if you go below milliseconds, you have subdiffusion. So that's the observation. Once again, it's interesting to see the comments. Again, they say, well, we have this superdiffusion. It cannot be explained by the simple model for the cargo motor complex along a single microtubule. We need something more to explain it. And in this paper, they argue that it could be that the microtubules themselves are moving, which is possible. They have some pictures showing that indeed some microtubules can themselves be transported. But the important point is that people think that to explain superdiffusion, you need something more. Actually, it's quite easy to find anomalous diffusion. You can find many mechanisms giving you anomalous diffusion. And all the argument that they give could be true. The motion of microtubules could contribute. You find also superdiffusion when you take ballistic motion on a branch network, for example. For some types of networks, you would find, again, this superdiffusion. But what I want to stress is that people were thinking that you cannot find superdiffusion just with the dynamics of the cargo motor complex. So of course, after this, you would expect that if I insist so much, it's because Sarah simulated this cargo motor complex with the model I just described, the explicit model. And what does she find? She finds some superdiffusion with an exponent close to 3/2. And if you remember the scale, so the transition is around seconds. So we are in the good order of magnitude. Above, well, we don't have networks, we don't have obstacles or nothing, so you find diffusion. That's what you expect. But here, so you have this superdiffusion, and it seems the 3/2, it's just by chance that we have this for biological parameters. Actually, if you change the parameters, you vary this exponent around 3/2. So it's not specifically 3/2, but that's it. So my conclusion is that actually you don't need an extra ingredient to explain this superdiffusion. Now we could go to smaller time scale, and there we must add another ingredient. We must add thermal noise. So we do add some thermal noise to the motion of our cargo. And then what do we find? We still have the superdiffusion, but then we have a crossover towards subdiffusion. And this occurs at, in the order of magnitude of 10 microseconds. So we are again in the good order of magnitude. And this we can easily understand because a motor, the speed of a motor is about 1 micron per second, and the size of the step, 8 nanometers. So it's about 100 steps per second, and this gives you a period of 10 microseconds this time. So it means that for time scales below this, the motors are just not moving. They are fixed. And so you have this cargo moving due to thermal activation, and it's just linked with this spring, so it's in a trap, and you find subdiffusion as expected. And then when you go beyond this time scale, then the motors start to move, and there you have some superdiffusion and some correlations between the steps. If you start to go on the right, it's more likely that you will continue to go to the right.
May I ask a stupid question? You probably said it, but I missed it. How do you characterize superdiffusion? What is that? So
we measure the variance of the motion, and we look at the exponent in the, if I go back here. So we define the variance like this, right? And we plot it as a function of this delta t, and we find an exponent. And if it's between 1 and 2, we say it's superdiffusion.
So that's the definition of superdiffusion?
Yes, yeah. Okay, there are also some other observations which were made that in the trajectory of these objects, we find some relaxation events which are quite well fitted by an exponential. And people said, OK, there is some elastic component there that is relaxed. And again, they would say it's because microtubules are bent and suddenly, yes?
How it comes in the model is superdiffusion. Why there is superdiffusion? Is it the time between reversal?
Well, it's because once you go in one direction, you somehow give some advantage to this team, and it will have a tendency to continue more.
Quantitative explanation?
Not really, yeah. Yeah, it's a numerical result, I would say. Okay, we can make some effective model by adding, for example, we could make an effective model by describing at a more coarse-grained scale, and we can put some persistence in the motion that would reproduce this, but that's not an explanation. It's just a way to coarse-grain the stuff.
With long-range memory?
No, just between successive time steps. OK, we can discuss this afterwards. So OK, to explain, this, they would think, again, that you need some extra ingredient, like having microtubules buckled that would release this elastic energy. But actually, we do find the same relaxation events. And this is just because when one motor detaches, you release some elastic energy. So you don't need, again, some extra explanation, and it's about the same. You don't see it, but it's about the same types of scales. So we don't have so many experimental observations, but we can reproduce this anomalous diffusion, this elastic relaxation events. But we have not answered why is it so? Why is there an interest to do so? So now we explored several things. The first one is, imagine you have a microtubule where you have a viscosity in the middle, and then we put some barriers just by increasing the viscosity in some places. That's a very idealized case. And we want to have the distribution of the times when you start here to go outside of the barriers. So when you have the same viscosity everywhere, no obstacles, you find that diffusion can be more efficient. You have shorter times here. But when you increase the effective viscosity in the barriers by 10 or 100, then you find that it becomes more interesting to have this persistent motion. So the idea is that in a very crowded environment, it can be more interesting to have this motion with more persistence, just to cross barriers. Something even more interesting is that, as I said, the two teams are different. They do not react in the same way to a change in the force or in the ATP concentration. And this is very important. It means that just by changing one external parameter, you can give the lead to one or the other team. So if you increase the ATP concentration, which is the source of energy, you could think if you give more energy, you will go faster. This is not the case, because depending if you have little or high quantity of energy, you will give more benefit to one team or to the other. So what you find is that starting from a motion which is biased to the right, you can go to a motion which is biased to the left. And your velocity does not increase with the ATP concentration. It decreases and increases again. So this is a first observation. Of course, ATP concentration you don't control in a cell, but in an in vitro experiment, you could vary it.
I didn't understand, is it because different motors somehow absorb ATP differently, or what? Is that,
yes,
the reason?
Their stepping rate does not depend in the same way with the ATP concentration.
For the different kind of motors?
For the different kind of motors. So some will live better with little energy, and some will go faster when there is a lot of energy available. So you change the balance. This is true also with the force. So one way to change the force is to say you change the effective viscosity around. And there, when you increase the viscosity, again, intuitively, you would say when you have more viscosity, the velocity decreases. This is not the case. You can speed up the cargo by increasing the viscosity. And you can also reverse its motion just by increasing the viscosity. So this can give ideas about many possible scenarios. For example, if you arrive in a region which is more crowded, it's like increasing this effective viscosity, and you can imagine that automatically it reverses the bias and you go somewhere else. This is our hypothesis, but these are possible due to this, this control that you can make. Just by one external parameter, you can change the bias of the cargo. So we think this could be one reason why it's what the life chose for our cells, is that it's a highly controllable system. So now we would like to to control this by in vitro experiments. There are discussions with a team in Dresden to try to make this. Until recently, it was not possible to handle mammalian dynein in vitro, but now people have understood how to do it. So it opens a field of all kinds of experiments that could be done. And we would expect now we can characterize each motor individually and make collective experiments. So this is very promising. There is still a question, what we do in vitro, how is it in vivo? We know there are differences, and it's still an open question to know how much you can transpose. But I guess we will learn a lot about these things in the coming years. OK, now I will go to the second part. I will go a little quicker. So we want to understand how we go from a single pedestrian, now human, to a crowd. So I focus on collective effects. I will show you one you know. When you are in a corridor and you have many people, spontaneously people form lanes. Here they even form 5 lanes, which is quite unusual. But you can actually form quite a lot of lanes. Here we had made a different experiment in a ring corridor. So at the beginning, everybody is mixed. And as soon as they walk, you form 2 lanes or 3 lanes here, or even 4. And you have transitions between these different regimes. Another situation where you have patterns is when you have intersection of perpendicular flows. So it depends if you go only in one direction in each corridor, or if they are bidirectional. I will focus on this case, where mostly it forms diagonals. But you can also, depending how it is organized, form roundabouts in other situations. So we focus on situations where we have unidirectional flow in both corridors. This has been observed in some experiments. Here you see the crossing part. The arrows are the velocities, and the color is a kind of density field which has been reconstructed. So you guess there are some kind of diagonals. It's more visible in simulations. This is one type of model. You see these stripes here in another one. And here an example where you see the formation of these diagonals. There were also some models for city traffic. This is in a Manhattan-like city where you have perpendicular roads. And it's very idealized because people go only east and north. But then they again find this kind of stripes. I don't know if you can see it. It's very small, the roads. But there are, again, some diagonal stripes. Here is one example where you have 2 groups crossing perpendicularly. Well, it's a bit difficult to see it on the flight. But I will show you one picture. Of course, when they enter, the intersection is very disorganized. But then when they go out, you have a tendency to form diagonals. So we wanted to understand this. This was mostly the thesis of Julien Cividini. And we collaborated with Henki Lost. Julien is now postdoc at the Weizmann Institute with David Mukamel. So we model, it's quite a robust phenomenon. We modeled it in a very simple way. We have cellular automata. The yellow people go up to the north, so they will be labeled by N, and they go just straight, and the blue go east. And the only interaction is when there is somebody in front, you stop. So it's a very basic model. Also, we consider only low densities where we are in the free flow phase. Something important is that when you have a cellular automaton, it's not enough to give the geometry and the roles. You must say also in which order you update particles. Here, I will consider 2 updates, which have the interesting property that you have no conflict. When you have parallel update, 2 people could want to jump at the same cell. You want to avoid this. So either we just update one family of pedestrian, then the other one alternatively. Or we use an update where people have like a label between 0 and 1 in their back, and they advance in the order of this number. You could understand this when you step, you have a cycle, and you could say that the one which is more advanced in this cycle will go first if there is a priority conflict. So we will consider these two. We could consider other ones. Now, if you look at the intersection, you find these diagonals. So you may be surprised by the size of the intersection, which is much wider than what I showed you with the pedestrians. But what is important, actually, is the number of people you meet in the intersection, and it's the density times the size of the intersection. With cellular automata, you don't want to freeze it, and it's freezing very easily. So we go at very low densities, and thus we take a very large intersection. For pedestrians, rho is much higher and m is much smaller.
So what is just you sort of put different configurations on the intersections on top of each other? What did you plot?
This is an instantaneous picture of the intersection. You have the orange people and the blue people.
But I thought you were doing at very low densities.
Yes, but it's a very large intersection. If you look, it's hundreds. So this is
it's mostly empty.
This is a zoom, you see.
I see.
It's not so full. So depending on the update, you have slightly different structures, but basically you have the same. You have diagonals, and maybe you start to see that it's not really diagonals. Actually, you have like two triangles here. You start to see them. If you look more carefully, so in the upper part, in this part here, you see that the two species are not the same. One species forms very dense and narrow diagonal, and the other one is more homogeneous. And it is the contrary in the other triangle. And if you plot 45 degrees diagonals, you see that it's not exactly the angle of these lines. So actually, if I exaggerate so that you see something, we more have a structure like this with chevrons. And if you use periodic boundary conditions, you don't have them. So it's really linked to the fact that some species enter in one region, and when they arrive here in this one, when they arrive here, they have crossed already, they have met already a lot of people, while the blue ones here, they just enter the system. So they are not, it's not equivalent. So to summarize, periodic boundary conditions, you have diagonals. Open boundary conditions, you have chevrons. And if you mix, you have only one angle. So we made some mean field approximation. And what is interesting is that we find the same patterns as for the particle model, but this allows to make linear stability analysis. So I will go quickly. It's quite technical, but you find the unstable mode corresponding to diagonals. You can do this for periodic boundary conditions. There you have only diagonals. But also for open boundary conditions. It's more tricky, but again, you find only the diagonals. So the chevron effect is really a nonlinear effect. Can we understand it? So as I said, there is an asymmetry in the way both species are organized. And this gave us the idea that this system can sustain modes with tilted stripes. So here is one example. The yellow, they form a diagonal, except here you have a kink, but it's associated with a blue particle. Now imagine the blue move, so the A goes here. And then at the next time step, the yellow cannot move the B because there is somebody in front. So the kink will move in this direction to the right, while all the yellow go up. So at the next time step, you have exactly the same structure. It just moved one step to the right. So you have like this some stable modes with kinks. And actually, in your simulations, you can identify such lines here. It's a bit small, but okay, you can find these modes. So now the angle is just related to the number of kinks that you have. And these kinks are related to the density of these blue particles. So you can have an estimate for the angle, which gives an upper bound, because it assumes the blue are homogeneously distributed. And indeed, it works quite well, at least for small densities. Now, how is it created, these modes? Now we must think in terms of effective interactions between two particles. So we'll consider two blue particles in a field of yellow particles going up and see what is the effective interaction. There is a whole field of literature with effective interaction. The most well-known is depletion at equilibrium. But there is also more recently more research in out-of-equilibrium system. You want to know the effective interaction between probes within a flowing fluid or something like this. But usually these calculations are complicated and you make approximation. For example, you assume that the perturbation of the density field around is just a superposition of individual perturbed fields, or you look at special cases where the probes are aligned in the flow of the fluid and so on. In all these systems, you are interested in effective forces, but this means nothing for a pedestrian. You have no forces. There was a model which was closer to our model, where they would have two biased particles in a particle model. The blue are diffusing, and the red, they have a biased motion in one direction. And numerically, they could find that there was an attraction between the probes, but it was only numerical. In our case, as we have a very simple dynamics, we can do things exactly. So we can, for example, if we have one blue particle, we can compute the average weight of this particle. Of course, it's exact, donc the theory and simulation match, no surprise. But actually, what the other particle will see is not the average weight. It's one particular realization of the weight. So you must go really to the microscopic structure and basically track when one blue particle is moving, all the cells that it occupied in the past, which are transported with the yellow flow. When it's blocked, well, then you have only one, it occupies only one cell, and then it goes on. So you start to see again this structure with diagonals and kinks when there is a yellow particle. And OK, you can describe this. You have two types of rows, either 2 points or 1 point plus a yellow. And you can write the master equation. If you assume you have another particle in this wake, you can write all the equations for the transitions between these states and solve it exactly. I passed this. And in the end, what you obtain is how long the second particle will stay in the wake of the first one. So if they were independent, no wake, it would decrease quite fast. And if you add the fact that the first one, if you simulate the microscopic model, which includes the interaction, then you find that it will decrease much slower, or even alternating parallel update is a bit a special case. It will stay forever. So you will find some modes here which are exactly invariant in time, and that can live forever for alternating parallel update. For the other updates, it will survive for a certain amount of time. So this is an example of an effective interaction that we can solve analytically. In the full problem, it's more complicated. The yellow themselves, they would be slightly organized, and the angle may be quite different and depend on the update, but we would expect that the order of magnitude of the angle would not be that different. And the angle of this wake at the microscopic scale is exactly the angle of the long-lived global modes. So there is really a connection. We expect that the angle would be larger for higher densities. And now we had some new experiments in Rennes in 2016, where we did a lot of these experiments, crossing flows, and we expect to have enough data to try to see this effect in more controlled experiments. Okay, now I had a third part, but I think I'm out of my time. So this will be for the next time. And thank you for your attention.
For the crossing pedestrian thing, do you have experiment with animals crossing instead of humans crossing? And do you expect your model to apply equally for animals?
So I have no experiment coming in mind, but this is something important for the modeling, the type of models that people do. There are some models for animals, birds, fishes, sheep, and so on. And usually some difference with the pedestrian models is that the animals, they follow each other more or less. You have only one population. You could imagine to have more, but usually in the models you have, it's more that people, the birds, for example, they would more or less align with their neighbors and so on, and you have these big clouds going around. While in pedestrian models, something important, and which makes it quite complicated, is that they don't want to go all in the same direction, and they know where they want to go. And this is a big difference in the models. So now, I don't know examples where they would really cross each other, but maybe it exists with different species, maybe, or something, but I have no example coming in mind.
You just said that people know where they want to go. How do you replace that in your model, with knowledge? And also, how do you make the people behave politely, your model behave politely, so that they don't bump into each other? Because there's a lot of intelligence in these people.
Yes. Well, in my example, it's a very simple model. I have two types of pedestrians, and some are going up and some are going to the right. So there I have only two destinations. When people make more Navier-Stokes-like models, instead of having, usually you would write rho of x and t. And if you deal with pedestrians, usually you have to add where they go. So you have one extra parameter describing this. After, it depends. Of course, it's more easy when you have a quasi-one-dimensional model. So for example, in a corridor, we had made a model for bidirectional motion. There you have two populations. But you could imagine in a shopping mall or whatever, big square, to have a continuous distribution of these things. So, OK, I don't know if I answered to your...
How would you explain that these diagonals form in that way and not the other one? I mean, it seems quite stupid, but...
You mean when the flow...
So pass?
In the example I gave, so they go like this, and they would form like this.
Why
or not like this.
Yeah, that's
I think it's really a matter of wake. If you have seen that when one pedestrian walks, its wake is somehow transported by the other species. And it's just geometric effect that it must go in this direction.
Thank you.
Okay, so if no more questions, then maybe we can thank again Cécile. Thank you.